I'm going to talk about progress and pitfalls in COVID-19 vaccine development. And this will be followed by Raghavan, who's going to give you more details on the vaccines themselves. Now, if we think about outbreaks, epidemics, and pandemics, what Shahid set up was a beautiful exposition on why we have had such an increase in the number and diversity of outbreaks recently. Whether it's trade or travel, or the fact that we live closer to each other, that we move around a lot, that there's been deforestation and climate change, the risk of epidemics has increased because of all of these. And the alphabet for outbreak causing agents goes from AIDS to Zika. SARS-CoV-2 is just one in this really long line of infectious disease agents that we've seen emerge and go around the world. Now, one epidemic that brought to the forefront was that it's important to think globally was Ebola in 2014, 2015. Essentially, with Ebola, it had been occurring in Africa since 1976, but in 2014, it came into the cities and affected 3 countries in West Africa really badly. Overall, only about 30,000 people were infected, which is a number that sounds very small today, but was huge 5 years ago. Out of those 30,000 people, about a third died. And it was estimated by the World Bank that the epidemic had cost about $4 billion in direct costs. So the actual costs of treatment. And overall, the cost was $54 billion. If we compare that to SARS-CoV-2, it's been estimated that the optimistic take is that it's going to cost $3.3 trillion. And the consensus is that it'll be somewhere around $26.8 trillion over 5 years. And the worst case estimate, which I was too scared to put on the slide, is $82 trillion. That is 5.3% of GDP to be lost over a period of 5 years. Now, the sad thing about Ebola really was the fact that there was a vaccine that had been sitting in a freezer for a really long time. And it took us a long time to get that vaccine from a freezer into a trial. So if you look at what happened, Ebola was reported towards the beginning of 2014. And WHO had its ethics committee report back saying that a vaccine trial with that vaccine should be done on the 11th of August. It took us till the 5th of November to decide that a ring design was the design that was going to be appropriate. But it took until the 23rd of March to actually start vaccination. And you can see when vaccination in the course of the epidemic started. The peak of cases was in November. By the time the vaccination was actually initiated, the epidemic had essentially waned. There were hardly any cases that were left. And if you look at what it took, once you actually had the vaccine going, the efficacy data showed that the vaccine gave 100% protection. But the total number of people who were vaccinated in the immediate vaccination arm was approximately a couple of thousand, and delayed vaccination was another couple of thousand people. It took us a few weeks to find out that the vaccine worked. So we started vaccination in March. By July, we had the results. Had this all happened at the beginning of the epidemic, imagine the number of lives and the morbidity that could have been prevented by being able to use a vaccine early. So this resulted in calls for global action, and WHO decided to prepare every year an R&D blueprint to prevent epidemics. It meets in December of every year and lists 10 infectious disease pathogens that are likely to cause outbreaks. And there's always an 11th pathogen, which is called disease X, which is the pathogen that nobody knows when it will come, where it will come from. Now, in response to these calls for action following Ebola, the Coalition for Epidemic Preparedness Innovations was set up, and it was intended to counter exactly what had happened in Ebola, which is not to have a vaccine that sits in a freezer and doesn't get developed because of market failure. So the idea of CEPI was, can we make vaccines against what we know are going to be emerging infectious diseases, or employ platform technologies so that we can make sure that when outbreaks occur, we will have vaccines that are ready to be used and further developed during those outbreaks. This should be a solution that is sustainable so that there is equitable access to vaccines for the populations that need it the most. CEPI was established in 2017, and its ecosystem is now a large one. It has brought together players in academia, in industry, in NGOs that are from around the world. Now, CEPI developed a target portfolio, and this consisted initially of 3 pathogens that were identified by its scientific advisory committee on the basis of the likelihood of causing outbreaks and the probability of success in developing a vaccine. The initial targets were Lassa, MERS, and Nipah. Chikungunya was not initially included because it was thought that there would be commercial interest in a chikungunya vaccine, but then after that program was pulled, chikungunya was brought back on, and Rift Valley fever was added when there was additional funding available from the European Commission. In addition to the pathogen-specific targets, CEPI also established in 2018 platform technologies for disease X, with the idea that these would be technologies that would improve the speed of vaccine development against unknown pathogens. Now, because CEPI had funded 3 disease X programs or platform technology programs, in January of 2020, when the sequence of the SARS-CoV-2 became available, CEPI immediately moved its disease X programs to develop vaccines against SARS-CoV-2. Subsequently, it has added other programs and now has 9 vaccine candidates that have been funded since January of 2020. More recently, CEPI and the Gavi Alliance have been asked to lead the vaccine pillar of the ACT Accelerator, which is the Access to COVID-19 Tools Accelerator. In addition to CEPI, which is a global organization in India, we have IND-CEPI, which has been established by the Department of Biotechnology last year to support vaccine development for emerging infectious diseases. There is infrastructure that has been established at the National Capital Region Biotech Science Cluster, or at THSTI, which consists of a laboratory and building out a BSL-3 facility. And the idea is to engage with CEPI on the global stage, but also to build public health preparedness in India. And this is being tested now through INCEPI and the National Biopharma Mission, which are funding multiple efforts to develop technologies and tools that will be useful for SARS-CoV-2. Now, the ACT Accelerator consists of 3 arms, which is vaccines, therapeutics, and diagnostics. And as I mentioned, CEPI and Gavi are leading the vaccines pillar. And the goal here is 2 billion doses of vaccine should be made available to the world by the end of 2021. This is the first program where there is a focus on quickly getting out a new vaccine to people all across the world. The Act requires countries to join this facility and agree to abide by certain principles that will enable equitable access to populations, prioritizing those who are in need first. Many countries have signed on to this program, and Singapore is leading an effort called Friends of COVAX progress, which has gone really well. And where we are today is, and not much has changed since a week ago, is that we have a lot of vaccines that are in development. It's been estimated that there are about 250 different projects with about 125 serious candidates. 17 vaccines, shown here in black, are in clinical trials and various stages of clinical trials. In India, we've had our first approval for human trials a couple of days ago, and this is an inactivated vaccine candidate being developed by Bharat Biotech based on a strain from the National Institute of Virology. Now that's progress. We come now to the pitfalls, and there are a lot of those. And I'll take you through. This is not an exhaustive list. If we sit down and list all the problems with developing vaccines, there will be many, many more. But I thought I'd highlight a few key issues. Now, the idea of doing trials, you need to be where the disease is to show that the vaccine can prevent disease. So if the disease moves quickly and your sites are fixed, how do you handle that? WHO has recommended the idea of mobile teams that have the capacity to do trials in various places, and maybe that will be a solution. It takes time to set up a clinical trial, and the kinds of places and the people who are experienced with doing clinical trials are very limited. So if you have 250 plus candidates and you have limited places where studies can be done, how do you decide which vaccine to test? It's estimated that phase 3 studies, which are efficacy studies, will require at least 30,000 participants for each vaccine candidate for a point estimate of 50% efficacy. Now, how many studies can you do at the same time that involve 30,000 participants? Chinese companies are leading efforts to produce vaccines, and they are having serious issues with finding enough patients or places where they can start to do these studies. So they're currently negotiating with South American sites where there is currently a lot of disease. WHO is working with sites around the world that are multi-country trial sites, and this is a program called Solidarity 3. Safety issues with the vaccine are particularly important to consider. We know that we would like to induce antibodies to the virus, but if you have antibodies that bind the virus without neutralizing infectivity, those can cause increased disease through increased viral replication or through the formation of immune complexes that can be deposited in tissue, and this can lead to inflammation. Antibody-dependent enhancement, which is a significant concern, has been described for another virus that was used in cats with an alum adjuvant. There's also a worry about using inactivated vaccines because both measles and respiratory syncytial virus inactivated vaccines have resulted in vaccine-associated enhanced respiratory disease because it skews the immune response towards Th2, resulting in increased airway dysfunction and delayed virus clearance. So this has been the mechanisms and the effectors for both ADE and VAERD are shown here. And this is something for which assays have been very difficult to establish. We have some learnings from dengue vaccines where this is seen, where some assays that measure both subneutralizing antibodies as well as look at whether you get an FC-mediated increase in viral entry have been established, and there are people now looking to see whether we can establish such assays for SARS-CoV-2. Moving to animal models, hamsters, ferrets, primates are all being used in various facilities. Hamsters get upper and lower respiratory infection, but no severe disease and recover quite quickly. In ferrets, you don't see lower respiratory infections. In primates, there's slow growth of the virus. So there really is no ideal animal model in which infection can be established and good challenge studies be set up. It does look like hamsters are best at the moment, but we hope that there will be improved small animal models soon. In terms of assays, most of the assays that have been established have been for binding, and neutralization is an issue because these assays need to be done in a BSL-3 and you can't have high throughput for these assays. Now, T cells have been shown to be important in certainly infected individuals, and what assay would be appropriate to use in a vaccine trial is still something that is unknown. So looking at the probability of success of vaccines, most of the candidates are spike-based, and there is a worry around whether that is really sufficient. We think that the correlate of protection might be neutralizing antibodies to the spike, but we actually don't know whether that is necessary and sufficient. Binding antibodies, we know, appear to be less common in asymptomatic individuals, and they do wane quickly. However, on the other hand, we see that T cell responses are mounted by most infected individuals, but most of these studies are small and of short duration. Safety also continues to be a concern. ADE, VERD are still theoretical concerns, but we have seen fever at high doses with multiple candidates. Now, a lot of people have been discussing whether human infection or challenge are needed, and there is a group called One Day Sooner that has recruited 20,000 volunteers to participate in human challenge studies. And there is a discussion around whether we should be thinking about controlled human infection models being part of the regulatory pathway for the development of vaccines. However, there are significant concerns around the unpredictability of severe disease and the idea that there is no rescue drug available at this time. Now, when we come to manufacturing, the entire production capacity of the world does not exceed 8 billion doses of vaccine. And we are talking about 8 billion doses of people needing to be vaccinated and maybe needing to be vaccinated by more than one dose. We also have no idea how long protection will last. So will this wind up being like an influenza vaccine, where you have to vaccinate every year? If that is the case, then we are going to need to ramp up capacity for manufacture hugely. And right now, we don't know which platform is likely to be successful. So much of the manufacturing capacity that needs to be set up will need to be set up at risk. The question also comes up of access. WHO has laid out 5 principles of transparency to improve efficiency and accountability. looking at public health need, looking at procurement processes to improve access, collaboration with everyone to accelerate and scale up, and using ethics to inform allocation strategies in order to ensure that people who need vaccines can get them. But that comes against a background of increasing vaccine nationalism. In the US, they've established Operation Warp Speed, which has selected 5 candidates that are expected to deliver vaccine for the US by the end of this year. But which vaccine will work, we don't know, and how will head-to-head comparisons be done? The National Institutes of Health have come up with the active model, which is to do a direct comparison of various platforms of vaccines using essentially shared infrastructure and shared resources so that all of the vaccines are assessed together. So we do need vaccines and we need them fast, but there is so much that is unknown. Our chances of success are uncertain, but based on animal studies, we are hopeful that it will be possible to prevent at least disease, if not infection. Our timelines for development and for delivery of vaccines are uncertain. There are a lot of challenges there. But for India, there is a ray of hope in that our vaccine industry leads the world in number of doses. So if it is an appropriate platform, perhaps when it comes to scale, we will be able to deliver vaccines not just for ourselves, but also for the world. So thank you very much for your attention.